Predicting T-cell receptor specificity is crucial for the development of immunotherapy, providing new opportunities and strategies for personalized cancer immunotherapy. The authors establish a TCR generative specificity detection framework consisting of an antigen selector and a TCR classifier based on the random forest algorithm. This framework aims to efficiently screen out TCRs and target antigens, achieving TCR specificity prediction. Cancer is the leading cause of death worldwide, with 20 million new cases and 9.7 million deaths in 2022. Traditional methods such as surgery and chemotherapy are effective in treating cancer to a certain extent, but they are often not effective in preventing the metastatic spread of the disease through disseminated tumor cells. Immunotherapy, harnessing the immune system to fight cancer, has firmly established itself as a novel pillar of cancer care. The specificity of T-cell receptor plays a crucial role in immunotherapy. T-cells can recognize and bind to antigen peptides from the major histocompatibility complex through the TCR on the surface to trigger an immune response to recognize and attack pathogens and abnormal cells. Researching the specificity of TCR contributes to the development of immunotherapy. However, various experimental methods used to identify the interactions between TCRs and peptides presented by MHC molecules have limitations, such as being time-consuming, costly, or technically demanding. The authors reviewed various computational models developed for TCR specificity prediction, categorizing them into three classes according to their working principle. The first class predicts TCR specificity based on three-dimensional structural information of TCR and PMHC. The second class is based on sequence information of the TCR and PMHC, utilizing high-throughput sequencing methods and single-cell RNA sequencing technologies. The third class combines the characteristics of the first two models, making full use of structure and sequence information to improve accuracy and reliability. The author's framework includes two key components: an antigen selector, which calculates and edits the distance matrix of TCRs between the target TCR and other TCRs in the training database, and a TCR classifier based on the random forest algorithm. The antigen of the closest training data is selected as the target antigen. The chosen target antigen and CDR3 are encoded by engram, and then the encoded CDR3 is used as input for the random forest algorithm. The authors present a machine learning framework for predicting TCR specificity using random forest models. The framework takes in TCR and antigen sequences as input features and outputs a prediction result. The model framework involves calculating the edit distance between TCR sequences, constructing a random forest model, and evaluating the model using k-fold validation. The edit distance calculation is a crucial step, measuring the minimum number of operations required to transform one sequence into another. The authors provide four formulas to calculate the edit distance, considering additions, deletions, and modifications. The edit distance is used to select candidate antigens for the random forest model. The random forest model is constructed using CDR3 and antigen sequences encoded by engrams as feature inputs and labels as model outputs. The authors provide a diagram illustrating the construction of the random forest model. The Gini coefficient is used to evaluate the decision trees, measuring the impurity of the dataset. For model evaluation, k-fold validation is adopted, dividing the data into k subsets, one for validation and the others for training. The authors also describe the VDJDB dataset, a comprehensive antigen-specific TCR sequence database, and considered 12 attributes valuable for TCR specificity testing. Finally, the authors discuss dataset preprocessing, detecting missing values in the database, as shown in Table 1. The table on page 3 of the research paper reveals no missing values in the dataset for the required attributes. A Spearman correlation analysis is conducted to identify collinearity among these attributes, revealing high correlations between MHC class and MHC B, as well as between J, SEM, V, SEM, and gene. To avoid collinearity issues in the model, it is necessary to exclude highly correlated data attributes from simultaneous use. Data categories for specific attributes are also analyzed, highlighting the importance of filtering out paired data that only selects the MHC category as MHCI and belongs to the human species. This is consistent with previous studies by Liu Tianqi's team, Rudolph, M.G., and Kim, S.M., who emphasized the significance of paired data and the role of alpha and beta chains in T-cell function. Based on this analysis, the pre-processing steps outlined include retaining specific attributes, filtering out data based on MHC, class, species, VDJ/DB, score, and rearranging paired data points into the same data point. The paper also discusses TCR visualization using principal component analysis, PCA, and TSNE, where three different distance matrices are calculated based on edit distance. Linear dimensionality reduction is applied first to reduce the size of the distance matrix, followed by PCA to reduce the dimensionality to 50 dimensions while retaining most of the information. The research explores the use of dimensionality reduction and clustering algorithms to predict the specificity of T-cell receptors, TCRs, based on their editing distances. The authors calculate editing distance matrices for TCR alpha, TRA, and beta, TRB, chains, applying TSNE for dimensionality reduction. Results in Figure 5 show clusters of TCRs with similar antigens having closer distances. Clustering algorithms like k-means and DBSCAN were evaluated, but direct application yielded poor performance. However, when clustering was performed on TCRs with dissimilar antigens, the effect was ideal, suggesting TCRs with close editing distances have similar antigens. The study achieved an average accuracy of 66.6% in predicting antigens using the nearest training data, with 30% of the data needing improvement. The calculation process is detailed in Figure 7. The research paper presents a novel model for generating target antigens through a distance matrix, leveraging the edit distance matrix of the T-cell receptor beta, TRB, chain between the target T-cell receptor, TCR, and other TCRs in the training database. This approach employs an engram encoding method for both CDR3 and antigen sequences, effectively capturing local structural information while retaining length distribution information. A comparative analysis of different encoding methods is conducted, including one-hot representation, improved one-hot representation, and word-bags representation. To address class imbalance, a random sampling method is utilized to select an equal number of negative and positive class data. Hyperparameter optimization of the engram model is achieved via k-fold validation, with the best accuracy attained using 250 decision trees in the random forest. The final hyperparameter design is determined by grid search, and the structure of a decision tree is visualized. This innovative approach contributes to the advancement of understanding T-cell receptor-antigen interactions and has implications for future research in immunology and bioinformatics. The authors present the results and discussions of their generative model for TC-rantigen prediction, demonstrating a minimal number of mispredictions in the confusion matrix, Figure 10. Performance indicators such as accuracy, recall, precision, and F1 score are calculated and shown in Table 8, indicating excellent performance. The ROC curve of the model is plotted in Figure 11, suggesting that adding a classifier for discrimination is quite effective compared to antigen prediction using a distance matrix. The authors discuss the limitations of classifiers, which require both the antigen sequence and the TCR-CDR3 sequence to make predictions, and their application scenarios are relatively small. In contrast, the proposed model can better adapt to more application scenarios. To compare the performance of different models, the authors calculated the accuracy of other classifier models and encoding methods through k-fold validation, k equals 5, as shown in Figure 12. From the graph, it is evident that ngram plus RF is the best performing classifier. Additionally, the authors constructed two deep learning models, LSTM and CNN, for training. The accuracy of the test set is shown in Figure 13. Overall, this section presents the effectiveness of the proposed generative model in predicting TC-rantigen interactions and compares its performance with other methods, highlighting its adaptability and potential for broader applications. The development of a T-cell receptor, TCR, specificity detection framework utilizing a random forest model is discussed in this paper. The framework consists of two parts, an antigen selector and a TCR classifier. The authors emphasize the importance of TCR-specific prediction technology in fields such as cancer immunotherapy and autoimmune disease treatment, highlighting its advantages over experimental methods in terms of efficiency, economy, and repeatability. The model outperforms ordinary algorithms, but it still requires improvement due to data scarcity and redundancy. To enhance the dataset, collecting more relevant data and using machine learning methods or data from multiple databases is suggested. Optimizing the model's algorithm for selecting target antigens is also proposed to reduce search time for some TCRs. The paper provides a comprehensive overview of the current state of TCR-specific prediction technology, citing various studies and databases, such as VDJDB.